Okie dokie. So we are actually going to start a new chapter, and that's going to be the chapter on meiosis. So we're going to do part 1 of the notes today, and we're going to have the homework of doing the chapter 10 summary and learn smart that's paired with this material, and that's going to be due by next Friday. It's going to overlap a little bit with us starting the next chapter or unit, but that is okay. And so our learning objective is that learners will be able to explain using visual representations or narratives as to how DNA in chromosomes is transmitted to the next generation via meiosis, followed by fertilization. And the reason why fertilization is important is because meiosis produces gametes, like sperm and egg. And sperm and egg each only have half the number of chromosomes as a human, for example. So you can't actually survive on just 23. You need 46 just in case one has a bad gene on it. So you need the other one to cover for it. And so fertilization brings an egg and a sperm together. So 23 plus 23 equals 46. And then you have a full set of chromosomes to be able to survive and function properly as a full example of a living human. It's the same in other animals that sexually reproduce. So meiosis is basically only for eukaryotic organisms that reproduce sexually. Not all eukaryotic organisms reproduce sexually all the time. Some reproduce asexually in cycles, and some reproduce asexually only. Now, that also means that prokaryotic organisms do not do meiosis. Remember, bacteria don't even properly do mitosis. They do binary fission, where they just kind of split apart by themselves and clone themselves. So there are a lot of different processes for producing more cells, and meiosis is often combined or compared to mitosis. So we'll talk about them in comparison as well, because we already learned about mitosis. So this do not question kind of helps you bring interest into why meiosis happens and what is happening inside. So this is a picture called a karyotype. And the numbers represent each chromosome. So there's chromosomes 1, 2, 3, 4, all the way to 22. And then there is a 23rd set of chromosome that is the sex chromosomes that determine the gender of an organism. Or in this case, since it is a human karyotype, it would determine the gender of this person, right? There are 2 X's, which means that this is a female human's karyotype. And if it was 1 X and 1 Y, then it would be a male human karyotype. Why are there 2 chromosomes for each number? Well, that's because one set comes from your mom and one set comes from your dad. So your mom provides you with 1 chromosome 1, 1 chromosome 2, 1 chromosome 3, 1 chromosome 4, all the way to 1 sex chromosome, which has to be an X from your mom because she only has X's, right? And then the other set, chromosome 1, 2, 3, 4, 5, comes from your dad. And your dad has an X and a Y. So he can give you an X or he can give you a Y. If he gives you a Y, then you're going to be a boy. If he gives you an X, then you're going to be a girl. Okay. So there's 2 chromosomes in each slot, basically, because one is from your mom and one is from your dad. When you compare the images of each of the 2 chromosomes, what do you notice about them? So when you notice the chromosomes for chromosome 1, they have the same shape, they have the same length, and they have the same genes on them. And the color here represents the genes. So it's kind of cool to see which genes are coded on which chromosomes. If you look at any of the pairs, you'll see that they pretty much look the same because they all contain the gene that codes maybe, for example, if chromosome 11 has a gene that codes for hair color, then both chromosome 11s will have the same type of gene. They'll both contain a gene for hair color. They might contain different hair colors in their genes. One might be a brunette gene and one might be a black hair gene, but they both contain a hair color gene, right? So that's marked by the color patches on these. Usually karyotypes are actually black and white, so you can't see the colors, but you can see the density and location of where genes generally are. Number 5, what is the term for these pairs of chromosomes? So when you look at the pair of chromosome 1, the pair is termed as a homologous pair. And homologous means the same, right? We have the same type of genes on the same type of chromosome, and they are both chromosome 1. And so in this organism, this is a homologous pair. Now, I'll go into the details of what sister chromatids are in the next slide. But just know that every single chromosome, if you want to split it apart, it kind of looks like an X shape, but it's not actually a crisscrossed X. It's more like if you took 2 straws and tied them with a rubber band in the middle. So if you were to separate them again, the vertical pieces actually separate instead of the crisscross pieces cutting into 4, right? So it's actually 2 long strands that are just stuck together in the middle. So if you look here, it says label these chromosomes, and they're already labeled. So when you look here, this is a homologous pair of chromosomes, and the blue generally represents even though it's gender stereotyping. The blue is generally representing a male chromosome or a chromosome that came from the dad. And then the red will be used to represent a chromosome that came from the mom. And as you can see down here, these two together are a homologous pair of chromosomes. Now, each chromosome has two chromatids, okay? And that means that you can pull this one and this one separate from each other. And originally they're connected in the middle by something called a kinetochore, right? Kinetochore sounds like connected. And so that's how you remember that these two chromatids are connected by a kinetochore in the center to make them into a chromosome. When you pull them apart, the center part is the centromere. So the centromere, sounds pretty similar. That is one single chromatid, okay? The chromatid, again, you can duplicate it to form it into a chromosome to get that X shape. The reason why it's marked as non-sister between these two chromosomes is because they are not on the same, between these two chromatids is because they're not on the same chromosome. If they are two chromatids on the same chromosome, like here, the right one and the left one, they're called sister or identical chromatids. Over here, some definitions for your term. So homologous chromosomes are chromosomes that have the same size or length and same shape, and they contain the same genes. Sister chromatids are when you split a chromosome down the middle and you get two pieces that have the same genetic information on them, and they come originally, like I said, from the same chromosome. Those are sister chromatids. Non-sister chromatids would be chromatids from the other homologous chromosome after they're split, right? Again, a chromatid is just that single line, not the X, right? The X is the chromosome shape, but when you pull it apart, that's a chromatid. But if you take a separate one and you pull that one apart, those two together combined or compared to each other are non-sister chromatids. Meiosis is defined as dividing cells from diploid to haploid. Diploid is double the amount. For example, in humans, it's 46 total number of chromosomes, to haploid, which is 23, to prepare for sexual reproduction, right? If you just took a human cell of 46 and added it to another human cell of 46, you would end up with a total of 92 chromosomes, which is too many. And that would create either huge mutations, huge disorders, or it just cause death and not even survivability, right? So that's why the chromosomes count is divided by 2 during meiosis to then be added together again in fertilization to total of 46 for a proper human number. Gametes refers to sex cells. So in humans, that would be, and in most eukaryotic organisms, that would be sperm and egg. And they are all haploid. Haploid, aka half, aka containing half the number of chromosomes. Somatic cells are any cell in a living organism other than the reproductive cells. So the other general term we use for somatic cells are just body cells. Again, haploid, we usually represent by N. It is a single set of unpaired chromosomes. So if they're haploid, that means you have half the number. Diploid is 2N, so there's 2 copies of each chromosome, so you have the full set of a living organism that you are supposed to have, especially for humans. Some plants actually have tetraploid, which is 4N, which is 4 sets instead of 2N, and that's actually normal for them. So there are differences between different types of organisms, but we'll generally use the terms haploid and diploid because that applies to us the best, and it's easy to explain. So you're going to answer the following questions. What is the purpose of sex or sexual reproduction? Well, the answer is to create genetic variation, right? What is sex? Well, sex is when you are able to join 2 gametes together in order to produce genetic variation in the next generation. Do bacteria have sex? Bacteria actually don't truly have sex the same way that eukaryotes do in creating new combinations of genes. They can do something that's similar, though, where it's called bacterial conjugation, where they actually meet up and swap chunks of their DNA, but they're not actually creating a whole new bacteria, a third bacteria baby out of the 2. Okay, so it's not really the same idea, but they can create new genetic variation by doing bacterial conjugation. So it's kind of similar, but not really. So what is an advantage of sexual reproduction? Well, you get a lot of genetic variation when you do sexual reproduction. And genetic variation means that there are chances of having higher survivability or lower survivability when natural selection comes into play. A disadvantage of sexual reproduction is that sexual reproduction requires a lot of energy to do, right, to create the gametes and then to fertilize the gametes, and then usually the female will be the one that is in charge of creating the embryo, right? That takes a lot of energy and resources to do. So sexual reproduction is a high cost mechanism, but it produces a high reward, which is a lot of genetic variation. An advantage of asexual reproduction, aka like cloning, is that it requires a lot less energy and resources and time put into it. And then a disadvantage is that since it's a clone, there are no genetic variations. So if a population was all based on clones of each other and a disease came and wiped through the population, all of them have the same immune system type, so they would all easily get wiped out together. So survivability isn't different there since there's no variation. Name an organism that asexually reproduces. We keep talking about bacteria, so that's a good example. Yeast is also a good example. Plants actually go through cycles. It's called alternation of generation. So every other generation, or based on the weather and resources available, they can asexually reproduce and produce spores that are basically clones of them, or they can do sexual reproduction on years where there's plenty of resources and plenty of water. Name an organism that reproduces sexually or asexually. Sorry, that was plants, because they can do both and they alternate their generation. Most of the organisms that we see on our daily lives are actually sexually reproducing. For example, any animals that you see, like cats, dogs, birds, those are all sexually reproducing animals. And here's another diagram. As you can see, meiosis is a process that produces gametes. And gametes, for example, are things like sperm and egg, and they are haploid cells because they contain half the number of chromosomes. They each contain, in humans, 23. And in this diagram, they each contain 3. When they are met together, or when fertilization occurs, a zygote is produced that has a diploid set, or a full set. And as you can see, 1, 2, 3, 4, 5, 6, there are 6 chromosomes in here. And it is going to become a somatic cell, or a body cell of the new organism that is forming. Over here, these are non-sister chromatids because they are not from the same chromosome, okay? But they are also homologous chromosomes to each other because they are the same type of chromosome, but one came from the sperm and one came from the egg. A diagram of the life cycle of humans. So adults are 2N. They are diploid, right? And females can produce eggs, males can produce sperm through the process of meiosis, where each are haploid, or represented by N here. Through fertilization, the sperm and the egg will come together and produce a zygote, which is now back to 2N. So cells are not often found in N. Most of the cells in your body are not actually haploid, and they're not actually gametic cells. And cells don't stay as gametic cells for a long time. The cycle of life doesn't have any phase that is staying in N for a long time, because once they're fertilized, they become back to 2N really, really quickly. And through the process of mitosis, of growing and making new cells, then staying as 2N, the baby will eventually grow and grow and grow and become an adult again. And the cycle will continue. So what are the 2 main purposes of meiosis? Again, genetic variation and reproduction. So I'm going to show some videos, and I will show the questions at the end again after I show the videos. But if you want to pause and write them down and give yourself a little bit of space to write down your answers while you watch the videos, that is a good idea. Again, I will show them here, and I will show them again after we watch the videos. The first question is, 1, what are the end products of meiosis 1? 2, after meiosis 1, are cells haploid or diploid? What are the end products of meiosis 2? After meiosis 2, are cells haploid or diploid? What are the end products of mitosis? How is mitosis different from meiosis? I want you to kind of describe what crossing over or recombination is, describe what independent assortment is, and explain how crossing over and independent assortment create genetic diversity. Okay, so I'm going to show you some videos now, and then we'll come back to the questions. So let me share audio.
Meiosis is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes. During fertilization, these haploid cells fuse to form a diploid offspring. Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA. Meiosis involves 2 divisions. The first division, Meiosis One, halves the number of chromosomes, and in the second division, Meiosis Two, the sister chromatids are split. The end result is 4 haploid cells. Now let's look at each of these divisions in more detail. Meiosis One begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres. The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs. During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent assortment. Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere. In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has 2 sister chromatids. However, we are separating homologous chromosomes into 2 different cells, so that each new cell will have only 1 member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, 2 cells are created, and each cell has half the number of chromosomes compared to the original cell. After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only 1 of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber. And in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Crossing over during meiosis allows recombination of genes between homologous chromosomes. This alters the linkage between genes on the same chromosome. Crossing over occurs frequently during prophase 1 of meiosis, when homologous chromosomes associate with each other in a structure called a tetrad. First, consider the possible inheritance of alleles for 2 genes on a particular chromosome when crossing over does not occur. Recall that 1 member of each pair of homologous chromosomes came from one parent. The other member came from the other parent. In this example, 1 parental chromosome had alleles capital A and capital B, while the other had alleles lowercase a and lowercase b. Without crossing over, each haploid cell produced will inherit a chromosome with the same combination of alleles as was on one of the parental chromosomes. Thus, the arrangement of linked genes is not altered in this case. Now, consider the case where there is crossing over in the region between the two genes. Since the crossing over is only between two individual chromatids, the possible haploid cells after meiosis have 4 different genetic combinations. 2 will come from the chromatids that did not participate in crossing over and thus will still have the parental combination of alleles, either capital A and capital B, or lowercase a and lowercase b. The other 2 will be recombinant, with either capital A and lowercase b, or lowercase a and capital B alleles. Thus, crossing over allows for the reassortment of linked genes.
So in this case, crossing over is creating these brand new combinations that have never existed before. And so it's kind of interesting to think about how basically, unless there are twins, every single organism every single organism that is produced through sexual reproduction is the first and only of its kind.
During Metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
So this is something known as independent assortment, where just the random assortment of the chromosomes while it's lined up on metaphase, either 1 or 2, causes the resulting gametes to be different and have a different combination of the mom versus the dad's chromosomes. So the previous video was recombination. This one is independent assortment. Now we'll do a comparison of meiosis and mitosis, if I have access.
Prior to meiosis or mitosis, DNA replication occurs. In both meiosis and mitosis, the nuclear membrane breaks down as the DNA organizes into chromosomes. In meiosis, chromosome pairs come together, or synapse, and crossing over occurs, resulting in mixing of the genetic information between the chromosome pairs. The paired chromosomes then align along the central plate of the cell and subsequently separate, one traveling to each end of the cell. In meiosis, a second division sequence occurs, resulting in 4 cells with half the number of chromosomes. Mitosis involves a single division sequence, resulting in 2 cells with no net change in the number of chromosomes.
Okay, so as you can see in this diagram, the mitosis process only has 1 split, whereas the meiosis process has 2 splits or 2 divisions. You can also see that in meiosis at the end, you have half the number of chromosomes that you started out with. Forget if they're duplicated or not in the X shape, but each chromatid is also a chromosome, okay? So in meiosis, the end result, you have twice as many cells, but you have half as many chromosomes in each cell. In mitosis, you have half as many cells as in meiosis, but you still have the original number of chromosomes inside of every single cell. And that's why that's basically cloning. So you should be able to go ahead and pause and answer these questions based on that information. And if you need to, you can go back and rewatch those clips to answer these questions. I will be looking in these when I check your notes for extra credit. So here are some diagrams that I'm going to ask that you draw out. So hopefully you have 2 different colors, whether it's a pen and a pencil, or it's a highlighter, or it's like a colored pencil, anything that you have that is a pencil and another color, or you can actually have 2 colors. So you're going to actually start out with one single cell that has 4 chromosomes in it. And one of them is a large chromosome and one of them is a small chromosome. Sorry, one type of chromosome is a large one and the other type is a small type. And so the large chromosome carries 2 genes on it, A and B, and the small chromosome carries D. The reason we didn't choose C is because it's really hard to see a capital versus a lowercase c when you're drawing, especially if it's handwriting. So we have capital A, capital B on the blue chromosome here. We have lowercase a, lowercase b on the red chromosome here. At the bottom here, we have a capital D on the blue chromosome, and we also have a lowercase d on the red chromosome. So basically, all of the chromosomes that came from the dad just happen to have the capital version of the genes, and all the chromosome genes that came from the mom are lowercase genes, or they're all the same types of genes. They just have different variations inside of their coding. Now, in Meiosis One, you're going to have these homologous pairs line up in the middle, like here in the metaphase plate, but they're going to line up as pairs. And so the pairs get separated. So on the left, we have the big red chromosome and we have the small blue one, and they get pulled to the left. And on the right, we have the big blue chromosome and we have the small red chromosome, and they get pulled to the right. After Meiosis One occurs, Meiosis Two will occur, where it'll be kind of like mitosis, where these chromosomes will line up in the middle in a single file line, and the chromatids will be split apart. And so here you will get a big red, a small blue, and a big red and a small blue. Therefore, you'd be getting capital A, capital B, lowercase a, lowercase b, and capital D. Lowercase a, lowercase b, capital D. On the other hand, over here, you're going to have the big blue and the small red. And in Meiosis 2, you're going to end up with results of having big blue, small red in both of these gametes. And that means you have capital A, capital B, and lowercase d as your 2 combinations. Now, independent assortment means that along the lines of the split, in Meiosis 1, the organization of the homologous pairs could have been different. And if they were different, we could have had the reds actually go together and the blues go together just by random chance of how they were situated together. And if that were true, then our end gametes would be capital A, lowercase a, lowercase b, lowercase d. Lowercase a, lowercase b, lowercase d. And over here we'd have all caps, right? So just by this one little change, you can actually already have genetic diversity, just because this is a possibility of having these kinds of gametes, and this is the other possibility of just changing one little thing, and that is due to independent assortment. Now think of the variation that could occur if there were 23 pairs instead of just 2 pairs. Here we're going to talk about crossing over, creating genetic diversity. So we know that crossing over is when pieces of homologous pairs that are overlapped when they're next to each other during Meiosis 1 are swapped with each other. So when you have these next to each other here, we're going to do crossing over, but we're going to do crossing over not between genes A and B. So that means we're going to cross over in gene D, which is on the small chromosome. So we'll just take any orientation. We're not talking about independent assortment for now, but just any orientation right now. And that just happens to be this one. And we're going to do a crossing over event in the D gene area. And that means that the blue and the red are going to swap this chunk that contains the gene on the chromatid that is next to each other. And after Meiosis 1, you're going to end up with the large red chromosome untouched, unchanged, and a small blue chromosome that actually has one leg with a chunk that is switched for gene D into a lowercase gene D. That means that when these split in Meiosis 2, you're going to end up with 2 different gametes, even though originally on the previous page over here, they were actually the same. So here, because one chunk on the right side leg has been swapped with a lowercase d chromosome gene, then you will have one that is haploid still, and you're going to have lowercase a, lowercase b, and a capital D. But on this side, you're going to have a, lowercase b, and a lowercase d, because part of this chromatid has been swapped with the other chromosome during crossing over in Meiosis 1. On the right side, we can see here that the big blue chromosome and the small red chromosome are pulled to this side. But remember that part of the small red chromosome has been swapped with the small blue chromosome on gene D because of crossing over. Now, when you perform Meiosis 2, you're going to get capital A, capital B, capital D for 1. and you're going to get capital A, capital B, lowercase d for the other. And this is hugely what genetic diversity is all about, because this produced 4 separate different haploid gametes just from this one little change. And that's kind of crazy. Okay. You'll notice here that I've labeled these as diploid up here because there is a full set of chromosomes. But right after you do meiosis 1, you go straight into haploid because now there's only 2 chromosomes in each cell. And it stays haploid because there's 2 chromosomes, albeit it's only chromatids now, but it's still just 2. So again, meiosis 1 is the division that makes diploid go to haploid, and then haploidy is maintained through meiosis 2. Let's do a crossing over with just gene B. And that means that the leg at the bottom of the large chromosome is going to switch with the one that is next door to it. And so once you swap the bottom half of the leg of the red and the blue large chromosomes, over here, you're going to perform meiosis 2 after you've done meiosis 1. And before you do that, you can see that right here, it is different over here now. Instead of having lowercase a and lowercase b, it is now lowercase a and capital B. But since we're not doing a crossing over event of the gene A or gene D, then those 2 remain untouched. So you'll have a combination at the end. On this side, you'll get lowercase b, and then a capital D. Over here, you'll get a lowercase a capital B, and a lowercase d. And that is the result. This capital B is a result of the crossing over. On the right side, you will notice that the blue has swapped indeed that chunk with the red, and therefore on the left side of this large blue chromosome, now we have a capital A and a lowercase b, which will bring in genetic variation because after you do meiosis 2, the resulting gametes now have a capital A, lowercase b, lowercase d option, and a capital A, capital B, lowercase d option. And if you look at all 4 of these, again, they're all different. So just changing one, just swapping one gene through genetic recombination already creates 4 separate different gametes. Now, I want you to show me a crossing over of genes A, B, and D. And I want to see what that looks like. As an example, I've started over here. So we're going to cross over and we're going to swap the whole bottom leg here containing gene A and B between the red and the blue here. And then we're also going to switch that chunk between the blue and red chromosomes that contains D. Notice that we never switch the ones on the outside, on the very left or on the very right, because they are not adjacent or not next to each other. So I want you to draw out the entire rest of the diagram here with the colors to represent what are the genetic combinations of A, B, and D that occur after you swap and cross over A, B, and D genes. Then I want you to show me, with independent assortment being different from the initial starting point of meiosis 1, how we could get different results when they're still crossing over of A, B, and D for these N4 and compare them to these 4. Okay, so we're going to use an organism with a haploid number of 3 now and a diploid number of 6. So we're starting with 6 to make another diagram of cells to model meiosis and how independent assortment can create genetic diversity. Okay, so we're going to do a crossing over event between genes A and B. And for this one, we're just going to keep all the reds and all the blues on the same side because, you know, by independent assortment and random chance, it is possible. So we're starting with our diploid cell, and we're going to swap pieces for A, and we're going to swap pieces for B, and it's going to swap right here in the middle, and then we're not going to swap D. Okay, so after meiosis 1, we will have a large red chromosome with a swapped blue leg for A. We'll have a large medium red chromosome with a swapped blue leg for B, and then we'll have an original red chromosome for D. And over here are combinations. Once we split the left sides and the right sides, we're going to have capital A, capital B, capital D. And we're also going to have lowercase a, lowercase b, and capital D. On this side where we had all the blue chromosomes, remember that the left leg of the large blue chromosome has been swapped with the red one for a capital A. The medium one has been swapped for a capital B, and then the D remains untouched. And so once you split them apart, left side, right side, down the middle, you're going to get capital A, capital B, lowercase d, and with a combination of lowercase a, lowercase b, lowercase d. And again, all 4 of these are different from each other. And that is genetic variation caused by independent assortment and by crossing over. here's another example where you're not going to cross over A and B, but you'll cross over genes D. I actually changed this from a haploid number of 3 to a haploid number of 4 and a diploid number of 8. I don't know. Actually, I take that back. Sorry, that was just me misreading this. We just added different genes here to make it a little bit more complicated. But it's fine because it's pretty much doing the same thing because we're only going to be manipulating genes D and E here. Okay, so we start with our diploid cell, and on our first big chromosome, we actually have 2 genes on there. We have A on the top arms, and we have B on the bottom legs. And then in the medium chromosome, we have D on the legs, and on the smallest chromosome, we have E on the legs. So when you split them after they've done recombination, we're going to have them do recombination between all the genes except for A and B, which means D and E are swapped. Okay, so that means that after D and E are swapped, the right side legs of the red are swapped with the left side legs of the blue, and we have some lowercase mingled into the red side now and some uppercase mingled into the blue side. When originally it was all capitals and it was all lowercase in the red and blue respectively. So now after you split them all down the middle, go through meiosis 2, you're going to have capital A, capital B, capital D, capital E as one gamete, and you're going to have capital A, capital B, lowercase d, and lowercase e as the other gamete. On this side, after you go through meiosis 2, you'll have lowercase a, lowercase b, capital D, capital E as a gamete. And lastly, you'll have lowercase all A, B, D, E for another gamete. So these are all the haploid gametes that are going to be different combinations, once again, like I've been saying, and that is the incredible version of, incredible part of recombination and independent assortment, being able to create so much genetic diversity in sexual reproduction. So what is different about metaphase and mitosis and meiosis? Well, if you look at Meiosis One compared to mitosis, the same number of chromosomes are actually lined up differently. So the chromosomes are lined up in pairs of homologous pairs, and they get pulled as full chromosomes to the side. So the chromatids are not separated during Meiosis One. In Mitosis One, though, they are separated, and they're in a single file line. Okay, that's why mitosis is more similarly compared to Meiosis 2, where there's a single file line. Although when you notice, Meiosis 2 has half the number of chromosomes as mitosis. Okay, so in Meiosis 1, the process is the same as in mitosis, but the chromosome count is different because it's already haploid here, even though in mitosis it is diploid. What is similar about metaphase and mitosis and meiosis? Well, like I said, all chromosomes are aligned in the metaphase plate in the center of the cell. Right, they're always aligned in the middle because metaphase, and then spindle fibers are attached to the kinetochores at the centromere, so they are attached to the middle of the chromosomes. And one more to reiterate, like I said, Meiosis 1 is not that similar to mitosis, except for these facts. But Meiosis 2 is quite similar to mitosis, except for the fact that there is a diploid in mitosis and then a haploid cell in meiosis. So here is a general table that is comparing Meiosis 1 and mitosis. Right, so in prophase 1, the homologous pairs are together, but in prophase and mitosis, there's no homologous pairs. In metaphase 1, bivalents or homologous pairs, another term, or a tetrad, you can call a tetrad a bivalent or homologous pair, are stuck together at the metaphase plate, whereas in metaphase of mitosis, it's just a single file line of all the chromosomes individually. In anaphase 1, when they're being pulled apart, the homologs are pulled apart rather than the chromatids like in mitosis. And in telophase, 2 haploid daughter cells are produced, and they're no longer identical to the parent cell, whereas in telophase of mitosis, they are 2 diploid cells, and they're extremely identical to the parent cell. Now we'll compare meiosis 2 to mitosis, and you'll notice that the prophase part is the same, where there are no paired chromosomes, right? So in metaphase, then, they're going to be lined up in a single file line, except in metaphase 2 of meiosis 2, it's haploid, so it's half the number of chromosomes lined up in that middle compared to the diploid number in mitosis. In anaphase 2, the sister chromatids become separate, as it does in anaphase of mitosis, and they move to the opposite poles or opposite ends of the cell. And then in telophase 2, 4 haploid daughter cells that are not identical are produced, versus at the end of mitosis where it is 2 diploid cells that are identical. Okay, so again, make sure you've answered the questions in the middle and that you've drawn all your diagrams. I will be checking that diagram in the middle that you were in charge of doing on your own, as well as the question answers when you turn in your notes for extra credit.